Now, all right. In the previous videos that you have come across and talking about the carbohydrate metabolism, biochemistry of carbohydrates, we stated the fact that there are various pathways that you need to consider while looking at the metabolism of carbohydrates. And we've looked at glycolytic pathway, and we said, remember that we stated that the end product of that glycolytic pathway is the pyruvate. Now, this pyruvate, remember we stated, if you know you've not watched the other two videos that I've done, just do well to watch the other two videos so you get to understand it much more better. Now, the pyruvate that is the end product that is formed, remember that glycolysis occurs in the cytoplasm. So be it in the liver, be it in the kidney, be it in the red blood cell, it occurs in the cytoplasm of those cells. Now, or in the cytosol if you might like. Now, this pyruvate that is the end product, remember, it's a 3-carbon what? It's a 3-carbon sugar, and it's 2-pyruvate that is formed from what? The glycolytic pathway. Now, we stated that this pyruvate, we have to be channeled into the TCA cycle. Now, that is the tricarboxylic acid cycle, which is also known as the Krebs cycle. But the pyruvate cannot enter directly into the mitochondrion. What will happen there is that this pyruvate is needed to be converted to what? To acetyl what? Acetyl CoA. Now remember that because we are having 2 pyruvates here, that what's going to be formed, the number of acetyl CoA that is going to be formed will be also be two what? Acetyl CoA. Now, is this acetyl CoA that now enters into the mitochondrion to go through the TCA cycle? Now, what's the importance or significance of the TCA cycle? Now, I'm going to break it down. One major importance of the TCA cycle is that it generates reducing equivalent, which are the what? Which are the NADH and the what? The FADH2. These are now channeled into the what? Into the electron transport what? Transport chain to generate what? To generate ATP by oxidative what? Phosphorylation. Now, this is just a quick breakdown. Now, this thing I said here now is very, very important that you consider it. Now, this is one significance of the TCA cycle. But please, the electron transport chain is not part of the TCA cycle. The TCA cycle is involved in oxidative phosphorylation. Now, it involves proteins or complexes that help to transport electrons to generate what? ATP. We are going to look at that. Do you get me? Remember, in before that video ended, the second video, you must have noticed that I said that one NADH is equivalent to what? To 3 ATP. So practically it's 2.5, but theoretically 3 ATP. But one FADH is significant or synonymous with what? With 2 what? With 2 ATP. Now, I needed to make the video very, very short. This video is going to be very, very short. So there are some things that I need to skip, but I'll make sure that you understand it properly. Now, this is one significance of the TCA cycle, generating what? Reducing equivalent. Now, another significance of the TCA cycle is that it serves as an intermediate in protein metabolism. So the intermediates that are formed in the TCA cycle are what? Are products from protein metabolism. Show you the get them now. Now, ladies and gentlemen, because we have said that, when we talk about protein metabolism, very fast, try forget talking about transamination and what? And deamination. So that means there are some proteins, there are some amino acids that undergo transamination to form intermediate in what? In the TCA cycle. And this intermediate, of course, can be converted to what? To glucose via gluconeogenesis. Remember, I told you about gluconeogenesis, that is the reverse of glycolysis, but the steps are not directly opposite of what? Glycolysis. Now, another thing I must bring out if I look at the TCA cycle, because the major aim now is that let's look at the TCA cycle. Remember that we stated something earlier on that the glucose, so what? So pyruvate is the glycolytic pathway, and we have looked at the 10 steps. Now, remember that we stated that galactose is an azote sugar. It's a 6-carbon sugar. It can also enter into the glycolytic pathway. Now, the pathway through which galactose enters into the glycolytic pathway, now that pathway is known as the what? As the LELOIR pathway. Now, this LELOIR pathway is the pathway through which galactose enter into the what? Into the glycolytic pathway. Show you the get them now. Now, how? First thing that happens in the LELOIR pathway is that is that this galactose is what is phosphorylated by who? Of course, phosphorylated ATP is involved to now form what? ADP. By who? By galactokinase. Show you this here now by galactokinase. So this galactokinase is a form of exokinase that catalyzes what? Galactose. And the problem here is that this galactokinase phosphorylate galactose in carbon number 1. So therefore, the product form will now be what? Galactose 1 what? Phosphate. Unlike what happens in the time of glucose during glycolytic pathway, that if I phosphorylate glucose, now form glucose 6 phosphate. But in this situation, now form galactose 1 phosphate. Now, what am I trying to do here? I'm trying to bring out the LELOIR pathway. How does galactose enters into the what? Into the glycolytic what? Pathway. Now, after that has happened, now this galactose 1 phosphate, what happens here is that there is a compound known as what? UDP what? Glucose. Now, this UDP glucose is uridine diphosphate glucose. Now, this compound here, what happens is that there's an exchange. There's an exchange between UDP and what? Between UDP glucose and galactose 1 phosphate, in the sense that this galactose comes to this. So this UDP is just like a vehicle that carries this glucose. So there's an exchange here. It will now give me what? UDP galactose. Show you the same in the sense that what will not be formed here will not be what? Glucose 1 what? 1 phosphate. Now, what's going on here is just a transferase enzyme. UDP, the enzyme that catalyzes reaction is what? Is galactose 1 phosphate UDP transferase. Show you the get am now. Now be the enzyme where they catalyze this particular word exchange. Now, when that happens, now you might ask me a simple question. Because it's a cycle, this needs to be converted to this so that the cycle can go on continuously. This have to be converted to this. And what reaction is that? Is what? Epimerase. Because if you remember, glucose and galactose are what? Epimers, to be precise. Glucose and galactose, in short, galactose is a C4 epimer of what? Glucose. So to convert to this guy is just an epimerization reaction that is done by what? An epimerase. Now, what now happens next is that this glucose 1 phosphate is being catalyzed by an enzyme known as what? A mutase. Remember, what does mutase do? Is a transfer kind of enzyme that transfer the phosphate from carbon number 1 to carbon number 6 to now form what? To now form, of course, glucose 6 phosphate. Now, this glucose 6 phosphate, of course, if you know that pathway, enter into the glycolytic pathway, and of course, continues with that pathway. So, what I have done here is to show how galactose enters into the what? Into the glycolytic pathway. And this pathway I've just shown to you now is known as the what? As the LELOIR pathway. This is known as the what? The LELOIR what? The LELOIR pathway. Now, fructose can also enter into the glycolytic pathway, phosphorylated by fructose kinase. The same way, but that pathway is not called LELOIR pathway though. So this one is very, very unique. Now, when there is deficiency of any of these enzymes, especially galactokinase, it will not result to lactose intolerance. How does lactose intolerance get to do with this? Remember that lactose is made of what? Of glucose and what? And galactose. So if you consume milk, and if you are consuming glucose and galactose, so if there is deficiency of this galactokinase, what will now happen is that the individual will